Alright kids, welcome to our final podcast installation. At the outset I would like to apologize to all my students, and especially, I think it was Mary Grace or Claire, but I can't quite place it, who explicitly asked whether or not we need to have the button pressed on the microphone for this to work, and it turns out, yes, we do. So we lost all of your contributions, including my warnings about what's going to be on the test, but at least hopefully my kids benefit from that. That said, let's get going. At the outset, I will tell you, with this podcast, you will need to still do some of your own research. You need to get out a piece of paper and make some notes and be able to write in order some of these systems that we need to trace the path of a certain thing going through it, and so forth. I will still try to hit as many highlights as I possibly can, though. So we started with the vascular system. And this is what you'll probably be faced with on the tissue level. And I want to remind you that arteries and veins both have three layers. The outermost is the one connective tissue. Inside of that is the smooth muscle, and this layer is the one that exhibits the primary difference between a vein and an artery. The artery is on the left. It's got a very thick layer of smooth muscle. The vein is over here. It's got a thin one, and that is due to the arteries needing to be able to sustain a much higher pressure because they're on the receiving end of the biggest push from the heart. The third innermost layer is called the endothelial layer. It's an epithelial type of tissue, and that just lines it. It's a single cell thick. I've included one extra image of this using a slightly different H&E stain, and that will that kind of just shows a stronger difference between the muscular layer and the connective tissue layer, with the muscular layer being more purple in that one. So again, I lost all your guys' contributions, but again, this diagram and the one for the arteries is your best friend. Learn all of them. Again, I encourage you to spend some extra time on the head. Internal, external, jugular, subscapular, axillary goes out to the arms, subclavian is the root branch that is coming off of the aorta, or rather, in some cases, even the brachiocephalic, I think is actually the case on the left side, or on the right side, but just make sure that you get all these guys and make sure you know them spatially related to each other. Some of these will be very hard to dissect to actually push in a pig, and you can build that into your strategy. But if we have the whole neck opened up and there's, if you look at this area, 6 veins staring at you, you need to know which one's which. And they'll be intermingled with arteries and so forth. Remember that a lot of the veins and arteries have the same names. So axillary up here, femoral or iliac down here, and so forth. I think you should also know what each of these things services, and I think this is a little bit important. Know what is a branch of the other. So, for example, in this upper part here, the axillary branches off of the subscapular, and the subscapular branches off of the subclavian, and there's other systems like that. So the example we had from our little quiz was the celiac branches off the aorta, but the hepatic branches off of the celiac. And so if the question is which vessel branches off of the aorta, in the case of arteries, it would have to be the celiac, not the hepatic. In terms of veins, please do not get the vena cava wrong. You should be able to trace that at least all the way into the heart. And so for the arteries, again, most of these match what you saw with the veins. This is actually where I goofed on the previous description. We've got the aortic arch, and there's 3 branches of that. You should consult your lab manual to get the specifics of that down, and keep in mind that this structure over here is going to include a couple of things that branch off in the brachiocephalic arch that if you're asked directly which one is what branches off of the aorta, for example, aortic arch, it's not going to be any of these finer things like the carotid artery or the axillary or subclavian, anything like that. So again, know what branches off of what. You should still know what goes through the heart and where. And then the arteries gives you a better opportunity to see what serves what. So again, know this diagram schematically in your brain. And now I need to emphasize, go to the old PowerPoints, go to the internet, and make sure you've seen pictures of these things, such that if you're faced with an actual dissected pig, and there's 3 or 4 red or blue veins, arteries next to each other, you can have a chance of dissecting which is which. Okay? So remember that the other major point in our discussion of the vasculature was that the fetus does it a little funnily. So remember that our blood supply is coming from the mother, so we have her exchanging oxygen from her blood at the placenta to the fetus's blood on the other side of the placenta, and that is coming up primarily through the umbilical vein. No. Through the umbilical, yeah. So the umbilical, through the umbilical vein. Okay. And so where the oxygen is in these vessels is a little bit funny compared to what it would be in the adult. So the oxygen-rich blood, again, is coming from mom, but it's kind of coming up and hitting the vena cava pretty early. And so the vena cava, which is normally returning pretty oxygen-poor blood in the adult, is actually delivering some of the most oxygen-rich blood in the fetus. On the next slide, we'll get to the various shunts. So ductus arteriosus, foramen ovale, and ductus venosus. I take it back. This is the slide. Hooray! Those functions, what 2 vessels they connect, and what they skip, are important for you to know. So I have the text on here. I won't read them, but make sure you can place that. So sorry for doing this out of order, but again, the blood supply is funny in the fetus. Remember, oxygen-rich blood is coming up the umbilical vein, okay, and it's using the ductus venosus to skip most of the liver, and that's going to put it into the vena cava coming up back to the heart. And the heart can then distribute the blood, as the previous slide said in the text, preferentially getting more oxygenated blood to the head, but I'm sorry, anyway, the usual distributive systems can actually still operate here. Okay, some of the blood still goes through the heart and gets pumped out, but preferentially this blood can skip the lungs and go straight out to the other organs too. Okay, so then we move on to the excretory system and urogenital. So you should know the very basic features of the kidney. So we're talking about the gross anatomy of this. So you should know the capsule is the outer coating, the medulla is the inner part, the cortex is the outer part, the papillae are the beginning of the collection tubes. Those end up emptying into the renal pelvis. That's kind of this broad thing. It actually looks like your pelvis in terms of the breadth of this structure, and ultimately dump into the ureter and go down to the bladder so that it can be excreted. And of course, from your vasculature, you would have had the renal artery and renal veins. Okay, so this is the actual excretion system. We've got the pig's butts down here. to give you a sense of orientation. Remember that the blood is going to come through the kidney, get filtered. The urine and the rest of the fluid to be excreted will go down through the ureters. They will meet up with the bladder on kind of the basal side of it, and on both sides. This stretched muscle here is the bladder, and then out the urogenital openings. So there's a couple different images of the female and the male reproductives in this podcast, and essentially you need to be able to trace the gamete from production to the end, to fertilization to implantation. And so with the female, how shall we do this? So the eggs obviously live in the ovary. They're going to come down through the uterine horn, otherwise known as the fallopian tube. In the next slide, I hope I have shown you. I'll show you a better uterus, but we'll see. One of the main distinctions we made in our class was between the pig's anatomy and the human anatomy. The embryos actually implant in the uterine horns or fallopian tubes in the pig. In the human, they implant in the uterus. And so that's specialized for their modes of giving birth. So the pig will have a litter, and humans will have one at a time. So one good strong structure in the center is good for us, whereas they have two whole horns of a strong uterus, and then only during delivery do they pump them into the uterus and kick them out one at a time. Okay, so again, if you're asked to identify these things, the ovary is the little darker bulb on the end, and then the uterine horns or fallopian tubes in the middle. They're kind of wind up together. The uterus hopefully will be in the middle, but it will not be impressive. And so this is evidence of that. Ultimately, sorry, I'm looking for the actual other structures. It's kind of hard to see the oviduct or uterus on the uterine horns on this animal, but you can see anyway that the uterus really is not that impressive of a structure, and that of course feeds down to the vagina, where we'll deliver and then of course both receive semen from the male. Alright, the male system has this a little bit more complicated. So to go from production of sperm, which happens in the testes, and that's that discrete lump here in this dissection. I think normally we left them alone, left them covered by the scrotum, but we'll see what's on the test. A reminder that the testes, which we first looked at, the seminiferous tubules, in our discussions of tissues in general. Remember that the interstitial cells here have an endocrine function and the production of testosterone. Okay, from the testes, once the sperm are produced, they are released and stored into the epididymis. That's this kind of hook shape here. It's got a kind of a coily, curdled appearance to it when we get it live. They're stored there until their scheduled release. They will travel through the vas deferens, which you can see here. And along the way, they're going to have different fluids contributed by three different structures: the seminal vesicles, the prostate gland, and the Cowper's gland, or the bulbourethral gland is another name for Cowper's. And it's important, you should look this up, to know the order in which these things are added. Okay. Once everything's mixed, they can make their way into the penis. I think we have a better picture of that in another image. But again, it's not something that's necessarily impressive from dissection from the inside. One other note of kind of just something that makes sense structurally. Remember that the testes are these really tightly coiled lumps of seminiferous tubules twisted and then bound by collagen and other connective tissues. So when we want to intentionally block the release of sperm, as in fixing men, what we actually tie off is so much more simple. It's just this singular duct, the vas deferens. So again, here's another dissection. You have a much better view of the bulbourethral glands, the urethra coming down from the bladder, yeah, so the penis is dissected away here. And again, it's not something you would necessarily be able to distinguish from the bladder or the urethra unless it's by position. And this is just, I like this picture because it's another couple looks at the different structures. Again, the testis is their own kind of lobe, the epididymis is surrounding around that, and the vas deferens emerging from that. Okay, and so one last view, just a close-up, especially because I thought this bulbourethral gland looked texturally different than what you see in the other images. It's just more looks. Okay, we then moved on to development. So these are the terms that you need to know. Remember, we start from a single unfertilized cell, which is not featured on this slide, but suffice it to say that it looks like any one cell zygote, except it has a nice sharp nucleus sitting right here. So once it is fertilized by a sperm, the nucleus dissolves, and it can begin its process of dividing. Obviously, the next step is 2. There's usually a series of synchronous divisions such that you get even multiples, exponential number of cells. 2 becomes 4, 4 becomes 8, which you may not be able to see in this, but maybe you could see here. And by the time you go through another division or 2, this raspberry-shaped structure is called a morula. I would stop calling it a morula after it gets to about 32 cells for the purposes of our test. So if you count this, you kind of see 1, 2, 3, 4, 1, 2, 3, 4, and there's probably about 8 groups like that. So that's about as many divisions as I would bother to call a morula. After that, call it a blastula. So even though this kind of looks qualitatively like the morula, I'd call that a blastula. And the blastula is just the ball of cells with the blastocell in the center of it, which will eventually invaginate, which you can see in these gastrula here and here, where basically what it is it's not really external force, but it's as if it is. So you start pushing from the outside, and actually this would have been the leading surface of that. This would have been the first structure on the outside that pushed inward. So like pushing your finger into your bubblegum bubble, you start forming a tube surrounding where your finger would be in that image. Okay. And so that process of folding in on your cells gives you the different germ layers, the ectoderm, which forms the hair, skin, teeth, endoderm, which forms essentially the guts and anything like that, lungs, stomach, gut, pancreas, so forth, liver, and then the mesoderm, which is going to form the muscles in the bone. In the next slide, you'll see a couple different stages. So again, you have gastrula here, and then just kind of the mature larva of the starfish here, in case it comes up. So the chicken, I believe I have 3 slides on this, illustrating just a couple different structures that you need to be able to recognize. First, we got the vitelline veins. That should be a nice landmark. You don't really see the notochord, but the notochord is an important distinguishing characteristic. Chordates, remember that not all chordates are vertebrates. All vertebrates are chordates, but things like lampreys, things that don't have an actual spine, vertebral column, are still chordates. Distinguishing characteristics of that: pharyngeal gill slits during development, a neural tube, and then the one other you can look up because I forgot it. The structures we can see on the chick embryo include the somites, which are these units of mesoderm that form alongside the notochord, and each one of those will become the muscle, connective tissue, and bone of a single vertebra. After that, you can kind of see we're on a ventral look at this guy, so we can see the three chambers of the heart of the chick coming together and looping out. That's a very distinct characteristic that you will very definitely be assessed on. And then you can see the outlines of the different lobes of the central nervous system. So there's a little bit of hindbrain you can see here, midbrain, the lumps in between, and then the forebrain. It's actually this whole mushroom shape, but these outloops are very specialized. They're actually what's going to become the eyes. The forebrain is essentially whatever's going to be left in that more anterior region. So as the embryo ages, you're still going to need to be able to parse hindbrain, midbrain, and forebrain. But this also gives you a look at some of the other things just a little bit older. So you can see the heart still loops out. You can actually see that the neural tube is empty. There's a hole in the center of it. We discussed that a little bit in class. You can see that the somites are forming together. And you can also see a couple of structures for the specialized sensory systems. So the eye has actually invaginated and induced another layer on top of it to give us the many folds of the eye. And you can actually start to see developing around the hindbrain the otic vesicle for the ears. And there's one more image next. So again, if you can find the hindbrain, the rhombencephalon, you'll see that the otic vesicle, where the ears are going to form, comes out of that area. I lied to Deshawn, the metencephalon's actually back here. I'm easily confused. Mesencephalon, mes for mid, becomes the midbrain, and then the telencephalon is made up of the diencephalon, the forebrain, prosencephalon. Why do you guys ever listen to me? The forebrain is made up of the prosencephalon is made of telencephalon and diencephalon. And again, forming off of the telencephalon, you see the optic cups for forming the eye. Again, you can see the heart looping out. You can see the pharyngeal gill slits. And if you squint, you can see the somites. Okay. We then moved on to the nervous system. And while this is a busy slide, don't panic. Remember that there's several key things that we're going to learn about the spinal cord. First is that input comes into the top. So with these nerves on the outside, they're actually a mixed nerve at this point, but at the top what's happening is sensory information is flowing into the top, the dorsal part of the spinal cord, and that can generally go anterior, or as I like to say north, back up to the brain to tell the brain what's going on. So far what I'm talking about is you touch something, you detect warmth, or you move your arm and you have proprioreceptive positional information, or you crush your hand or something like that. Those sensations are afferent with an A, and they're going to, they're the input, and they go in the dorsal side. The efferent, the ones that cause an effect, the motor neurons, the somatic nervous system, tend to happen on the ventral side of the spinal cord. Okay, so You can tell the ventral side by this cleft here. And if you really want to parse, you can say that some of the more, the autonomic system, the systems that aren't necessarily controlled by conscious thought or reflexes or anything like that, but really breathing, heartbeat, that sort of thing, those kind of nerves or nuclei happen in these more lateral positions. So we've got input on the top, output on the bottom. So if you tell your arm to move, this is where the signal is being sent from your brain down to your arm. Then the other thing, and I can touch on this in the next slide too, is the general structure of white matter versus gray matter. Remember that white matter is white because when you fix the tissue and you examine it histologically, what you see is the fat from myelin, which insulates the axons. And those axons are the ones that run from your brain down the length of the spinal cord. And so when we take a cross-section across the spinal cord, what we tend to see is just a little dot that's the axon, surrounded by a gaping hole that's kind of light in color. And each one of those is an axon running the length of your spine. Those myelinated nerves are concentrated on the outside of the spinal cord. This is the opposite of the brain, essentially. The cell bodies are concentrated in this butterfly shape, and so that contains the bulk of the inner neurons, glia, and the sensory neurons. But again, sensory tends to be on top, efferent tends to be on the bottom. But again, cell bodies in the butterfly shape. We can kind of see that histologically. When you go through it, there's a diagram in your book that's very helpful. But the afferent stuff comes in the top along this nerve and using these nuclei. The efferent, the ones that cause an effect, a motor effect, are down here at the bottom. And again, the cell bodies all live here. And if you zoomed in really close to this, you would see that it's mostly axons, which we've cut clear through, surrounded by little circles of myelin to insulate the electronic impulse. And again, if there's any kind of processing that needs to happen on a reflex or something, what it's going to encounter is an interneuron. It'll be in here, and it'll determine whether or not there's enough electronic inertia or enough signal coming from the afferent stuff to decide whether or not to pass along the signal and cause a motor reaction. The other part of the central nervous system is obviously the brain. We do parse it further and further down, but this gives us the structures that we're primarily interested in. The brainstem has many parts that we will discuss. The cerebellum is responsible for a lot of your motor processing. Again, I'll remind you that in humans, this is an extremely large structure. It might be the reason we have big brains in the first place, because it's something that we had to develop a very big cerebellum to accomplish this complex task of walking on 2 feet. And that might have just dragged along the rest of our brain evolutionarily into being big. The benefit of that is the cerebrum, the structure derived from the telencephalon, is responsible for all of our higher order thought, emotion, planning, that sort of thing. So that's good, that makes us smart. So when we chop the brain sagittally, which is right between the eyes and going backward, we can see all the structures that we need for our test, including optic chiasm, which is responsible for taking input from the optic nerve, processing it. And then, actually, so we already talked about the cerebrum and the cerebellum. From the rest, I'd like to work from the posterior forward. So you have the spinal cord. That'll be something that dangles and is kind of wussy. The medulla oblongata is, for our purposes, principally just where that gets thicker. And the medulla has a set of functions. They're primarily autonomic. I'll let you read on the details of this, but let me just give you the general rule that, well, two general rules, that the higher you go up toward the brain, the more sophisticated this, the behavioral or the responsibility of that structure, the more complicated the task becomes, the more you move to the front. Okay? So, medulla handles things like your heartbeat. The pons, I want to point out, and this is a special structure, if the best way to look at it is actually ventrally, so looking up at it from the bottom, and that makes it clear that the pons is what actually connects the cerebellum. It kind of loops around and kind of just ties the whole thing back into the midbrain. And its appearance is much like the cerebellum, and it's kind of got these striations on it. So it's kind of got a homologous appearance as well. Again, that's much more, it adds some more sophisticated things, and because it ties into the cerebellum, it has a lot of these kind of motor functions with it. So, along with some other ones that you would simply have to memorize, in my mind, there are things, let's see. Oh, I thought I had it. So really some more of it's the beginning of the some of the inputs from some of the cranial nerves, sending those things out, eye movement. Here it is, equilibrium, that's what it was. So again, it's kind of taking that kind of processing from the cerebellum and then moving it out to wherever it needs to go, either back down the spinal cord or back up to the cortex for more processing. The midbrain, mesencephalon, again, it's an integration center for some of these cranial nerves, so vision, hearing, motor control, so special senses. And then it starts adding some of these kind of conscious functions, sleep, wake, arousal, and I think, don't quote me on this, do check your book, has to do with some of these more kind of primitive program behaviors like sexual arousal. That's good, especially considering, or at least it makes sense, especially because it's juxtaposed next to the thalamus, which again gives us a little bit more fight or flight, whether or not you're awake, that sort of thing. And that's kind of a nice little lump there in the middle. And then the cerebral cortex is this outer layer, including all the gyri and sulci, the dips and the lumps that we customarily think of a brain as. There's one other important, well, there's two other important structures, and they're right on top of each other. The ventricles contain the cerebrospinal fluid, important for balance and homeostasis and just in general taking good care of your neurons. But then this white matter streak right here, the corpus callosum, allows communication between the two hemispheres, and also is kind of a track so we can send information back down out through the spinal cord. And if it's white, that means that it's myelinated. It's an insulated connective tissue. This is essentially the fiber optic cable center of the brain. I just included this slide both to point out the cerebellum and then the striations on the pons, like I talked about before, but also both to label these four lobes. Frontal, I think that's easy enough to remember. Temporal, remember your temples. Occipital, I think of this as occidental, meaning the west, and to me, well, maybe I learned on a brain that was pointed the other way, so west was over there. I don't know, but it makes sense. And parietal's up to you to remember. And the reason I bother to go through those is because the bones of the skull will probably be named similarly. It's a good way to just to correlate those two, is to get another image of it. After that, we have the specialized sense organs, and again, I want you to be able to trace the path of something through the whole structure. So in this case, it's the passing of a photon through the whole eye to where it's detected and sends the signal onto the brain. And I'll do it in 2 slides, and I'll also at the same time talk about the 3 tunics of the eye, the 3 layers. And the eye is essentially a sphere, so you have to think of spherical layers, and we cover those as outermost to innermost. If I have to do that piecemeal, I'm sorry, but we can start at the outside anyway. The outermost layer, the outermost tunic, is the one from the fibrous tunic, the connective tissue tunic, and that means everything on this is going to be tough. The fibrous tunic, you need to remember, has 2 components. The sclera, the famous white part of the eye, covers about 5/6 of the eye. Another 1/6 is the cornea. The cornea is a specialized part of the fibrous tunic in that it is clear. There's one other important fact for our lab. The cornea, along with the lens, bends the light and puts it back to the back of the eye. The cornea, not the lens, the cornea is responsible for bending most of the light. So in terms of the optics, if this were a compound microscope, the thing doing most of the work, which is essentially the objective lens, is the cornea. The big one's the cornea. The cornea does 75% of the work. Okay? We think of the lens, but the cornea is more important for that purpose. Okay? I'll get to the other layers on the next slide, but from the outset, this is a good time to say, as we start tracing our photon coming into the eye, it's going to pass through the clear cornea. Depending on the dilation of the muscular iris, it will pass through the pupil if it can, if the pupil is big or small, and then move on to the structures internal to this. The only other thing to point out here is the iris is actually the part that carries the color of a person's eyes. And remember, the iris is controlling the size of the pupil. I think I may have included another slide after this, but this gets at the structures that a photon will pass through pretty well. So the photon comes in, again it hits the clear cornea, which is part of the tough outer fibrous tunic. It's going to pass through the anterior chamber filled with the aqueous humor, go through the pupil, so the pupil's this kind of gray shaded area here. The iris is blue in this case, and it's been cut away revealing pink. So again, we go through the pupil, we hit the lens. The lens is shaped by the ciliary bodies on the side and the muscles therein, so they'll actually squish the lens, which is much more flexible than when we dissected it. It was not flexible when we dissected only because it was fixed with paraformaldehyde. But you do remember there's that, a lot of people call it a marble in there. That's the lens, and it's directly up against the iris and the pupil. This will do the fine work of actually focusing the light, so we have inputs coming from the brain to these ciliary bodies to tell the eye how to shape the lens. After it hits that, it'll go through the very clear vitreous humor, that's the jelly that fell out and or squirted you. It'll come to the back of the eye, to the general region called the macula, and I owe Katie an explanation. Macula, or macula lutea, is actually the reflective surface on the back of the lens, back of the retina, or back of the neural tunic, I guess. And when we dissected it, this was the blue part. Essentially what it is it reflects the light back through the photoreceptor layer a second time to kind of amplify low-level light. So a lot of species have this blue macula lutea layer, and For the humans, we don't really have it. So when we take a photograph and light shines off of the back of our eye, all you see is the red from the blood supply, whereas if you do it with an animal, you see a reflection of the macula. So that's what's responsible for the night eyes. A subset of the macula is the fovea, and this fovea is a small pit at the very center of the eye, which is kind of the hot spot for where the lens is focusing the light, and that contains the preponderance of cone cells. Remember, cone cells are the photoreceptors that are responsible for passing on color information surrounding the fovea and the surrounding area. So the fovea is in the center, and the concentric circles around that area, you'll have the rod photoreceptors, which are responsive to general light intensity, and they are much more sensitive. So to finish that up, after the photoreceptors in the neural tunic, in the retina, these are synonyms, after those cells have been activated, they send their electronic impulses down the optic nerve and back toward the optic chiasm and the brain. Okay, so to review the three layers of the eye at one time, remember the outer, tougher layer that you can, scratch a little bit, not have things go squirting out into space. The fibrous tunic contains the cornea, that's the clear part, and the sclera, that's the white part. There is a central layer called the vascular tunic that contains all the supporting vasculature that keeps the eye oxygenated, as well as choroid, which is generally responsible for secreting juices and things to make the humors happy. I believe the ciliary bodies are also part of the middle tunic. Then you have the innermost, which is just this thin layer of pigment, and the retina. And the retina, again, contains the photoreceptor cells. So it's called neural because photoreceptors are a kind of nervous cell, and they are actually the ones who send along the electrical impulse down the optic nerve. There's two disease states that we generally talk about. Remember that the lens, when that gets cloudy, that's called a cataract, and when the ciliary bodies don't do their job of maintaining the pressure of the aqueous humor that's in here, there ends up being too much or too little pressure on the cornea, and when you get that little puff of air at the eye doctor, the amount that your cornea compresses tells you whether or not you've had that condition. If it's out of balance, is called glaucoma. There was one other big thing that we covered in our experiments, and that was darn it, I'm forgetting the phenomenon, what it's called, but after images. If you stare at a particular color, the color photoreceptors, your cones, which are specialized to detect either red, blue, or green light, can get tired. Before I go on, remember that the way that you perceive light, you interpret a signal that is somewhere that activates both the blue and the red photoreceptors as magenta. You would interpret something that activates both the red and the green photoreceptors as yellow, and so forth. Blue and green make cyan. If you have all three photoreceptors activated, you see white. If you look really closely at your computer screen, at each of these you'll see that only the blue and red pixels, parts of a pixel, are turned on here. All three colors are turned on here. Only the green here, only the red here, only the blue here, and so forth. And the tricks of how that works are not necessarily important, but there is a logic to what you will see if you tire out a specific photoreceptor. So if you sat here and stared a good long time at only the red portion, you would bleach out the pigments that in the red photoreceptors that are responsible for accepting that red light. They would essentially get tired, and they would no longer fire as red. So if you looked at a white screen, after looking and burning out your red photoreceptors, essentially what you would see is only the blue and the green impulses coming through, so you would interpret it as cyan. So it's kind of an inverse after image that's left once you stare at something. And I would encourage you, as an exercise, to go through, well, if you stared at the green and then stared at white, what would you see? If you stared at blue and then stared at white, what would you see? And then if you really want to trick yourself, say if you stared at blue and bleached out your blue photoreceptors, then looked at magenta, what would you see? And then talk to your classmates. The other specialized sensory organ is the ear. And at the outset, we need to say that the ear does two things. The ear is responsible for your vestibular senses, and that is the only function for which we should talk about the semicircular canals. So let's just get those out of the way. It's called vestibular because those three semicircular canals connect to the vestibule. This contains fluid and a set of hair cells. And what this detects is rotational or linear acceleration or angular acceleration are the three terms that I've heard for it. And so if you think of this structure, there are three semicircles all oriented orthogonally. So one of these will take the x-plane, one of these will take the y-plane, and one of them will take the z-plane. And if you rotate this, say if you moved your head, because we're kind of looking at this from the top, if you moved your head just to the left or right, essentially you would accelerate the fluid that's in this canal in this direction if you were turning your head to screen right. Or excuse me, screen left. Sorry, not an actor. Anyway, if you turned your head this way, the fluid in it essentially would lag behind, if we get into physics, and go this way. Anyway, the movement of that fluid, the turbulence from that, is detected by a set of hair cells in the vestibule here. And the same is true as if you rotated your head straight down, the hair cells at the base of this would detect that. And if you moved your head kind of cattywampus, that other X, Y, Z coordinate will be interpreted by the hair cells down in here. So that's all fine and good. That is one sense that is derived from the inner ear. So in terms of hearing itself, what this image lacks is the outer ear, which we'll call the pinnae or the auricle. Make sure you know how those are spelled. The sound impulse will then travel through the external auditory canal, hit the tympanic membrane, otherwise known as the eardrum, and then start to transmit the sound mechanically from that to the inner ear. And in a minute I'll have you pay attention to what's air and what's fluid. So once the sound has hit the tympanic membrane, that vibrates and it moves the malleus, think of a mallet, this is also known as the hammer, the incus, which I don't know if there's a good way to know, but that's also called the anvil. Mallets tend to strike anvils. That then transmits the sound impulse, again mechanically, through what I call the stapes. Others call stirrup, and others pronounce stapes correctly, which impacts the oval window, which is a membrane on the inner ear, and that transduces the mechanical force from the sound into the vestibule. Now, this is where you need to stop and just not be confused. Though the vestibule and the hair cells in there are responsible for vestibular sense, that is the 3D positional information of where your head is, even though the same kind of cell, hair cells, is performing that function, the structure responsible for detecting the movement of fluid via hair cells that is responsible for our sense of sound comes from the cochlea. And I can't bash this over the head enough, apparently. The cochlea, this conch shell shaped thing, which Dom will tell me again and again, you can remember that this is responsible for hearing, because if you hold a seashell up to your ear, you can hear the ocean. Just remember, once we get the sound transmitted mechanically through the oval window, though it's connected to the vestibule, it goes straight to the cochlea and gets interpreted as sound there. From there, with the electrical impulses from stimulating the hair cells, the impulse goes out via the auditory nerve, the vestibular cochlear nerve. There's one other structure in here called the eustachian tube. That's down here. This connects to the inside of your mouth, and it's responsible for equalizing the pressure on the inside and outside of the tympanic membrane. So this is famous for your for relieving pressure mid-flight when your ears pop. So both the external auditory canal and the internal auditory canal are basically air-based structures, whereas inside the vestibule and the cochlea, there's that contains fluid. So just to make it clear, when you take off in a plane, there is normal atmospheric pressure on this side of the auditory canal and on this side of the auditory canal. As you get higher and higher, the pressure drops out here, but because the eustachian tube is almost always closed, this pressure remains at 1 atmosphere, the same pressure that we had on the ground. When you have a nice yawn or you chew some gum, what happens is this is able to open and the pressure is allowed to flow back out down through your mouth and equilibrate. So that relieves the pressure on this side of the ear, which was otherwise pushing out and making you uncomfortable. Same thing when you descend from flight. Now that you've yawned once mid-flight, you have low pressure built up in here from your high altitude. When you start sinking, the pressure from the higher pressure from lower on the ground, lower to the ground, starts pushing inside on this, so you have to yawn and chew your gums again, in which case the higher pressure from the lower altitude comes back up and starts pushing back on the other side of the membrane equally to the outside. I was going to say that my kids wonderfully took care of the skeleton, but apparently I failed to hit the right button on my microphone. I will again emphasize, though, at least again to them, that what you need to know are the major bones. So if you're in the skull, it's the things in your book. They're listed. If it's the axillary system, it's the major bones, major set of bones, and let's see. I think I'll go through it just a little, but again, I'm not an expert. What you need to pay attention to in general is what bone will be connected to what. So we'll say, what does it articulate to? And what kind of joint do the two bones form together? And what you should have is a couple of examples of each kind of joint, so you understand which ones they are, and even if you're faced with an unknown connection, you'd be able to judge based on what you know about that joint. Sorry, there is one set of things to know about bone tissue on the tissue level. Know that the osteon is the basic unit of the living bone tissue, so it's this whole circular unit. The canals, well, let's back up. The blood supply comes from this central Haversian canal, so that's got the blood vessel in it. A lacuna is Latin or Greek for lake, and somebody thought that looked like a lake, and that's where the osteocytes live that actually build the tissue back up. There's a separate set of cells, the osteoclasts, that tear down the bone tissue and remodel it. But the living osteocytes live in lacunae for plural. And then canaliculi, or canaliculus is the singular of this, are these channels that exchange the fluid in between the two osteons and the blood vessels. I'm sorry, not the two osteons, the two osteocytes and the vasculature. So again, pay attention to the major bones. It's good for your learning to be able to tell what one's left, which one's right, what's facing forward, and so forth. It'll also help you identify what the bone is. But consult your lab manual for the bones that make up the pelvic girdle. There's a set. Know the big bones, the clavicle, scapula, humerus, ulna, radius, tarsals, metatarsals, and phalanges, what they all refer to. So that's the appendicular part, that's the appendixes. The axial consists of the skull, the vertebral column, sternum and rib cage, as well as the joints that actually fuse the spinal cord to the pelvis. A couple of key points about this. There's consult your lab manual, sorry. But a number of true ribs that are connected via cartilage directly to the sternum. Then following those sets, there's I think 3 that are connected secondarily to the cartilage of another rib, but do not connect directly to the sternum. Those are called the false ribs, and I think the false ribs include these free-floating ones at the back that are not connected at all. Remember, the rib cage fulfills one of those basic functions of the skeletal system, which is protection. The rest of the skeleton also provides support and a means for locomotion. Should have been talking about locomotion when we were talking about the appendicular skeleton. We'll get to the skull in a sec. There are there's 3 different kinds of vertebrae that you need to know, and just study up. Cervicals on the top, thoracics in the middle, and then lumbars at the lower. They have an increasing heft as you go down, and they have characteristic appearances of the spines on the outside of them, the spinous process. And then there's a couple of specialized at the top here to provide the joint for your head to actually rotate on its hinge. So the atlas and the axis are the first 2 cervical vertebrae. Then there's a specialized ones for connecting down to the pelvis, including the sacrum and the tailbone, the coccyx. If you get a chance, sneak into lab and or find some information on which of the spinous processes belong to which. The skull, again, look at the list of bones in your book, and it will not extend beyond that. Some of the key ones: the frontal, the sphenoid is this, it only shows a little bit on the outside, but it's kind of a small joint in front of the temporal bone. Again, temple is responsible for this name, temporal bone. Remember that the mandible's on the bottom. Maxima, which max, think of it as higher, is the top jawbone. And then there's a series of bones inside that make sense considering where they're at. Nasal bones, the nose. The lacrimal, which has to do with tears, is next to the tear ducts and so forth. Moving on. At the base of the skull, you can see that some of those bones are more irregular than they look at the outside. And there's also a few holes that you need to deal with, including one for the spinal cord to go up, foramen magnum, as well as some of the vasculature. So we have carotid, jugular, and I don't know what these interior ones are, but that should about cover that need. I can't stress this enough. You need to know at least a couple of examples of what joints, of the different kinds of joints. And there's a couple of different levels of information on this. There's whether or not they're fibrous, like the joints in the sutures of the brain, whether they're cartilaginous, So you need to appreciate that level. Whether they're, I think synovial's the other one, which means they're free floating. But then mostly in the synovial are the ones we think of as joints that are going to be free moving, and they have increasing or decreasing levels of movement. So the ball and socket is the most free moving. That's an example like your shoulder. Then there's let's see. So some of the movements in your hand have another axis move along. There's let's see. There's specialized ones with complete rotational movement, such as at your head, the atlas and axis. There's some where two planes just kind of meet up, and they can kind of slide in that plane. So that's the example in your shoulder at the clavicle. There's I cannot tell you the difference between a hinge and a saddle joint. You can see based on the structure of this that they're only going to slide past each other really in one plane, and I think it's probably I think kind of schematized here. So we're talking about in the hand, there's a little bit of flexion laterally in the saddle joint, whereas in the hinge joint, like your elbow, it's strictly one plane. You can't wobble your elbow like you can your fingers. Again, both because it's late and because I'm not the best expert on these, you need to go through and make sure you appreciate what kind of joint each that you might encounter is. And I think that's it, guys. Good luck. I'm sorry that this carried on for an hour and that I lost your guys' stuff. Also, dig this out along with the other podcasts 3 or 4 months from now. See how much of this you can help yourself retain. Maybe it'll come in handy someday.